So in this video, we are going to now think about if we had isolated a specific protein through our column chromatography or isoelectric focusing, whatever, we would like to sequence that. What protein is it? What is the order of the amino acids? We're going to talk about two techniques. One is this Edman degradation. I left this in here actually so you could read it, but let's just take a look. Essentially, what we are doing in that process is we are taking this phenyl isothiocyanate right here, and essentially what we're calling labeling, we're binding it to the N-terminal of our polypeptide. And so that's what we see right here. And then we can release it. We can break that one amino acid that was the N-terminal off without messing up or hydrolyzing the rest of the amino acid sequence. And so now I can do some analysis and figure out what amino acid that is, and I have a new N-terminal. So if you think about it, we could keep doing this in series. So I labeled the first one shown in blue. I pulled it off, and I'm going to analyze and figure out what amino acid that is. Oh, now I'm going to label the new N-terminal, and I'm going to pull it off and figure out what amino acid it is. And so we can essentially snip off these amino acids one by one to figure out the order, which all sounds great, but we run into some problems because it's not 100% effective, and really it degrades over time. And so we really can't sequence these polypeptides that are longer than, say, about 50 amino acids. So if we want to sequence the whole protein, first we're going to have to break it up into peptides that are 50 amino acids or fewer. And then we can analyze each of those, but we're going to have to figure out in what order they went together. So we are going to cleave those with a chemical or with a enzyme. And we have lots of different options. So cyanogen bromide will always break the polypeptide at the carboxyl side of methionine. Or trypsin will always break it the carboxyl side of lysine and arginine. What else do I want to look at? Chymotrypsin, carboxyl side of tyrosine, tryptophan, phenylalanine, leucine, and methionine. So we have these things that we know where they will cut the polypeptides. All right, so let's think about how we would do this. I have this long polypeptide. I don't actually know the order of, and that's what I want to figure out. So I am going to treat it in one sample. I've got one sample. I'm going to treat it with trypsin. And when I do that, I end up with these two pieces that then I can use my Edman degradation to figure out the order of these. So that's how I know that this is threonine, phenylalanine, valine, lysine. Now I'm going to take a new sample, and this time instead of trypsin, I'm going to treat it with chymotrypsin. That's going to break it in different places, so I'm going to end up with different fragments. And then, y'all, it's essentially a logic puzzle. So, for example, I'm going to come over here and I'm going to say, I've got two alanines next to each other. And I've got this other piece, but I don't know if this other piece, does it go here? Is it after the lysine? Or does it go here? Maybe this piece was the first piece, the N-terminal, and it goes before those alanines. But my clue comes from over here because it was broken up differently, and I know it goes alanine, alanine, tryptophan, but wow, hey, lysine is to the left. So I know that this piece must fit in here. So that is the basic idea of using these overlaps to figure out the overall long sequence. All right, excellent. I hope that you found that interesting and fun. And that is the end of this video.